And we will move on to our next speaker, who's one of our sponsors, Natalia Mitin from Sapere Bio. We are obviously extremely grateful for your support and really look forward to your talk.
Thank you for having me. All right, good. Good morning, everybody. So a lot of themes. Thank you for having me present our most recent work. An amazing conference, as always, and there is definitely an advantage of going on day 3 because a lot of themes that our work touches on has already been introduced, so it makes my life that much easier. So we already heard about the importance of the immune system in general driving and general aging. We heard about the fascinating case studies from the proactive medicine clinics. It is really amazing to see the sort of the raise of awareness and the value in the proactive medicine and sort of the desire to stay healthy longer and studies that are initiated in that setting. And our work centers around measuring cellular senescence and key markers that affect the ability of the immune system to clear senescent cells. And so we think of it sort of as this immune aging system. And if we can understand that system, we can understand how to regulate aging trajectory in humans. So I will show you how we measure these measures, and I will show you that these measures are extremely heterogeneous in the healthy population, even in participants who are in their 20s. And then I will show some of this super early but really exciting work on showing how we're measuring these measures in the clinic. Cellular senescence really doesn't need an introduction, but as we all know, when you have exposures to various stressors, subcellular changes can happen that would lead to cellular senescence. Cellular senescent cells become permanently arrested and resistant to apoptosis, and typically immune system is able to clear these senescent cells. In the last few years, various cellular subtypes in the immune system have been identified that can serve that function of clearing senescent cells. So the most studied ones are natural killer cells in the macrophages, and then some work has also shown that iNKTs and neutrophils can also clear senescent cells. In the last 12 or so months, there were several studies, some of them are back-to-back in the same journal, showing an important and essential role of adaptive immune system in this process. So in particular, this paper from the lab of Manuel Serrano and Ines Marin showed that CD8 T cells, CD8 positive T cells, are essential for immunosurveillance of senescent cells. And so the model we are focusing on is measuring what we call 2 domains of aging. So we're measuring cellular senescence that accumulates with aging, and we're measuring the ability of T cells, so the T cell function, ability of the immune system to clear these senescent cells. As I mentioned, these processes normally exist in a balance, and then as you age or you have sort of different insults to the immune system that erode the immune system, the processes become deregulated. So we believe that understanding the system, understanding how these domains function and how they're regulated can be used to both monitor the aging, predict early decline well before any chronic diseases present themselves, and can also be used to identify interventions that can bring the system back in a balance and decelerate aging. So the 2 key measures of immune system aging are cellular senescence that is measured by P16 expression in peripheral T cells. The assay to measure P16, I'm sure as you're all aware, but the assay to measure P16 in humans was developed in the lab of my co-founder, Dr. Ned Sharpless. Ned's lab identified expression of P16 in peripheral T cell lymphocytes as a measure of pathological senescence in humans. And while cellular senescence is organismal, the seminal work from Matt Yusesveda then in Laura Niederhofer's lab have shown that induction of cellular senescence in just the immune cells has a capacity to cause accumulation of senescent cells in distant organs and promote aging. And so we at Sapere Bio, we built on Ned's early work and developed an analytically and clinically validated assay that has precision and reproducibility that is essential for the clinical use. We have also developed a sample collection kit that again is needed to a preservation system that is needed to allow clinicians to send the samples for analysis. I know those details might sound boring and not scientifically interested and probably not something NIH would be interested in funding, but trust me, these things are extremely critical when you talk about clinical measures, because in any clinical study, if you're looking at prospective measures, it's essential to know how precise your measures are and whether results are valid. And so we spent a lot of time to make sure that those measures have been validated and maintain a strict quality control, and that's why we're very confident in our clinical data. So to capture the T cell function in the immune system, we developed a measure that we are calling ILS, or Immune Longevity Score, and I will address that in a minute. And so this shows you so we think of these as two components of the immune system that we're focusing on, cellular senescence and T cell function. So this is the framework that we use. This is a framework that they use. So there are patients that will have normal immune senescence and normal T cell function. Then there could be patients that have accelerated cellular senescence while still maintaining normal T cell function, and vice versa, normal senescence and accelerated and deregulated T cell function. And then the last cohort where both metrics are abnormal. So the work I will present today focuses on how those things are sort of present in the healthy longevity cohort and how we use them in the clinic. All the work is derived from a healthy cohort of two types of patients. So we have a community cohort where we enrolled 250 patients, and these are participants, they're participants, they're not patients. These are participants that are interested in longevity research and longevity interventions. And another cohort, a physician-guided cohort with 232 participants, where these are the patients that seek care in the proactive longevity clinic, and are interested in that. These are the biomarkers that we identified that are essential to capture this process. I don't have time to go through sort of why those biomarkers were chosen, but happy to discuss later. But I will briefly introduce all of them. Some of them might be familiar to you and some of them might be new. So the P14ARF is sort of more of a well-known marker. So it can set up cellular senescence via 53p21 growth arrest, and it can also induce T cell exhaustion via indirectly in T cells. LAG3 is a very well-known marker of T cell exhaustion and the newest and hottest immuno-oncology drug target. Molecularly, the way it works, it modulates T cell activation by binding directly to T cell receptor. CD28 is a very well-established marker of T cell proliferation. CD244 is probably the least known one. It's a marker of CD8 memory cell health. And so when CD244 goes up in memory cells with aging, they become apoptotic while increasing proliferation. They undergo terminal differentiation. And also very interestingly, the CD244 seems to be working as an autophagy inhibitor directly through Beclin-1 complex. And we heard yesterday about the essential role of autophagy in maintenance of CD8 memory cells. So immune longevity score. So as the markers, the immune markers that I showed you are all markers of T cell function. So as you imagine, they don't work independently of each other. So what we did is we took those markers and we put them into the model to account sort of for the interaction between the variables and potential antagonistic effects. And we used as an endpoint, we used the measure that is measure of whole immune function called immune resilience. That measure captures the ability of immune system to withstand antigen challenge and suppress chronic inflammation. So it's not just a measure of T cells, it's a measure of the entire immune system performance. That metric has been reported by Sunil Ahuja in a recent paper. So here's the pairwise interaction to show you the complexity of the model. Again, happy to discuss later. It's a complicated slide. So as you can see, the model also included, I forgot to mention, but the model also included gender and chronological age to account for the fact that females tend to have a better immune system. So as you can see that, for example, for LAG and CD244, not only that the interactions are complex and often not linear, but there seems to be a change from detrimental to beneficial status in the same marker as with a chronological age. What we noticed interestingly, which I thought was super fascinating, that change happens to occur around 50, so around the middle age is where all of a sudden the levels of this marker go from detrimental to beneficial and vice versa. And so while these markers were measured in T cells and isolated T cells, because the model was based on the immune system function, on the functional outcome, as you can see, ILS correlated not only with the T cell function, but it correlated with changes, with age-dependent changes in all the subsets of immune system, including RBC and platelets and monocytes and B cells. So it is not a T cell specific function. More importantly, both ILS and P16 correlated with signs of early dysfunction. So remember, this is a healthy cohort. So these patients did not have chronic diseases, traditionally defined chronic diseases. So we used lab measures to sort of identify kind of this early signs of decline. And so as you can see, like for example, in the pulmonary, both ILS and P16 correlate with the pulmonary function and so on. But there were some exceptions. For example, in the insulin resistance seems to be exclusively associated with P16, and we see it in other cohorts too, whereas the hormonal function seems to be exclusively associated with ILS and not P16. Probably not surprising, so the better immune longevity is positively associated with estrogen levels and negatively associated with testosterone. So probably not surprising given, again, that females are reported to have a better immune system, but we thought that was really interesting finding, and you can screen grab and look at it more details. So let's see. So here's what the measures look like in the population. So again, this is a healthy cohort of between 20 and 90. This is the cellular senescence, and this is the immune longevity score. You can see the huge heterogeneity in every age group, essentially, especially for ILS, huge heterogeneity within the population, even in people in their early 20s. So it's really not an aging phenotype, it's the function phenotype. So this is how we're using this in the clinic. We are partnering with select clinicians in longevity practices where this is used to monitor patients and interventions. We're collaborating with a number of clinicians on clinical trials where these measures are used in the clinical trial to try to understand the intervention's efficacy and then potential subcohort analysis. And we refer to these measures under the umbrella of commercially, I refer to it as Sapere X, and this is the Sapere X report for one of the study participants. So it shows cellular senescence and immune longevity on the population level, and it shows longitudinal changes with sort of certain treatments. Again, Sapere X has been analytically validated and to have the precision and reproducibility that we need to perform this work. So this is the framework I introduced in the very beginning, and I want to get back to it to discuss the four groups. So again, these people have balanced immune system and cellular senescence. These people have accelerated cellular senescence, accelerated T cell dysfunction, and both. And so now if we look in the cohort, in our healthy cohort, here's how these cohorts are distributed. Here's how these phenotypes are distributed. So in early ages before the age of 45, majority, 50%, but still majority of people have sort of both domains are balanced. You can see, quite a few people who have immune T cell dysfunction, and then some people start to accumulate cellular senescence. There are very few people who have abnormality in both domains. As you move into the middle age, there is now a sharp decrease, doubling of people who have cellular senescence abnormality. and more importantly, a very large increase in people who have abnormality in both domains. Not surprisingly, as you move into the population that is older, over 65, now you have enormous increase in people who have abnormality in both domains. So in the last few minutes left, I would like to show you some case studies. Again, don't really have time to go through them as a case study, but I just want to show you how we use them and what the results are. So these patients have high cellular senescence. So they have normal immune longevity. You can see they have normal immune longevity in their high 70s, but they have accelerated cellular senescence in this one as well. So they have really high cellular senescence. These people have low immune longevity score, so they have normal cell senescence, population average cell senescence, but low immune longevity. And these people have both, so they have really high cellular senescence and really low immune longevity. Patients who have high cellular senescence, that's where clinicians will start to look for the causes of cellular senescence. Is it stress, and what can we do about it? Patients who have unregulated ILS, do they have acute infections? Did they have recent vaccinations? Do they have chronic inflammation that might be affecting it? And those patients might then start to progress into this last bucket because of upregulated immune senescence. The patients in this last cohort are the most troubling because these are the patients that now have both domain downregulated, both domain deregulated, and without the intervention, they will not be able to revert to normal under normal circumstances, so the intervention is needed. And the last two slides, Morten, very sorry, I want to show you responsiveness to the intervention, which is a key piece of anything that is an aging biomarker, right? So can we drive, other interventions that can bring those processes, can we drive them back towards the balance? There's a number of promising interventions that have already been mentioned. They can potentially be used here, that can potentially work. One important note that I think is really critical, and to Yusin's point, is that the interventions don't have to be senolytic in mechanism. Because of the biomarkers of aging are connected, any intervention can be applied. If it changes cellular senescence, it will be expected to change the aging because cellular senescence is a hallmark of aging. So it doesn't have to be a senolytic. And basically this is what I said. So all we're trying to understand here is, can those processes be reversed? Can we see that change with an analytical precision that is critical for clinical decision-making? And then what are the interventions? So the registry is completely agnostic to treatments. We're just collecting information, trying to make a short list of interventions that can make that change. And as you can imagine, the results are yes. So here are the results. So these patients have, you can see the change in, so cellular senescence didn't change, but ILS doubled. It now became normal. And this, again, cellular senescence didn't change, but ILS increased from single digits to 60. And then these case studies, I'll just go through them quickly. ILS did not change, but cellular senescence, even though it was normal, at least age-appropriate, cellular senescence decreased. So this is what we're doing with this work. This is where we see this, our work fitting. Here's my contact information, and Amy, who's in the back, her information. Feel free to find us. Feel free to email us. Happy to take questions.
Thank you so much for a great talk. We have time for one question. Maybe I can ask, is this available globally, or will it become available?
So it will be, depending on the definition of availability. Commercially to clinic, it will be available in the US. As a collaboration, yes, it's available globally for the clinical trials.
Okay. Quick question.
How much is the added value of having this immunosenescent? Okay. Yeah, thank you for your presentation. Mm-hmm. As a clinician, so we are interested
Yep.
of the added value.
Yep.
In addition to the regular risk factors,
mm-hmm.
do you have any quantitative feel of this?
Yes, so this does not correlate with, so this is an early predictor. So these people have no comorbidities. There is no way to see the early dysfunction. These are the people that have dysfunction that you cannot see clinically. And that's why clinicians like using it, because when we talk to clinicians, they say, this is a healthy patient, everything is fine, then they do this, then they go back and say, yeah, you know, maybe they should decrease their stress. Yeah, maybe they should not over-exercise. It allows you to see things that you cannot see clinically before it's too late.
All right, thank you so much, Natalia. That was really amazing.
Sure.